So the teaching objectives of this session is, they're basically 5. First one is, what do EVs have to offer as therapeutic agents? And then what are the 3 strategies of EV engineering? What are the current applications of EVs in drug delivery? What are the current applications of EVs in regenerative medicine? And finally, what are considerations taken when applying EVs to therapy? Before I begin, I do want to emphasize that this is a very basic, very broad survey of EV applications in therapy. So forgive me if this is common knowledge to all of you, but the hope is those of you who aren't necessarily involved in therapeutic application of EVs are empowered, encouraged, and hopefully excited to examine the role of your EV population potentially as a therapeutic. So there are 2 overarching applications of EVs in therapy. The first one is to use EVs as shuttles for drug delivery. Those include the delivery of chemotherapeutic agents, protein therapy, vaccine delivery, and the like. And then the second is the one that's been the topic of my research for many, many years, the application of EVs as mediators of cell therapy in the field of regenerative medicine. So there are 2 ways that we can modify EVs. The first one is to modify the cargo, and the second one is to modify the surface. In one case, you're modifying the signals that reach the target cell, and then the other, you're modifying the delivery and the tropism of this EV. So the first one is co-incubation, which is very simply incubating the drug with the cell, the parent cell, and it's a very passive method. It's technically simple, but the challenge with this is it's very inconsistent as far as loading efficiency is concerned. And many of the factors that were discussed earlier by my colleagues, including hypoxia, confluency, and other cellular factors really do impact how much of the target drug is loaded into the EVs. The second approach is through EV transfection. So you take EVs that are already purified, and then you use some sort of transfection reagent to shuttle the drug product into the EVs. Of course, that introduces several different challenges, including any toxicity that might be associated with the transfection reagent in the first place. If you remember, one of the advantages of EVs is that they're biomaterials and they don't necessarily carry the same toxic profile as something like lipofectamine, which is significantly more toxic in vivo. And then finally, if you're lucky enough to understand some of the pathways that drive the secretion of therapeutically potent EVs, you can engineer the cells themselves to either modify an aspect of the EV or modify the entire cell secretome to produce more therapeutically effective EVs. Of course, the problem with that is anything that involves genetic engineering comes with its own safety considerations, especially if you're using things like lentiviruses or agents that might not necessarily lend themselves well to clinical application. And as I mentioned before, the second way by which EVs can be engineered is through modification of the EV particle itself. And one general consideration is you always start with EV engineering starts with the cell, or the choice of an EV always starts with the cell. And so if you're looking for EVs for targeting a tumor, you're either looking for EVs that derive from macrophages or dendritic cells or immune cells, or you're looking for EVs that derive from the tumor itself. Presumably those will be better uptaken. But then beyond that natural targeting, let's see if I can use this mouse here. Can people see my pointer? Okay. So one way to modify the surface is through chemical modification using long-chain fatty acids or polyethylene glycols, which have now become highly specialized. There are several different types of different conformations and branched, and each of them endow the EV with a certain level of circulation time and tissue targeting. The other way that you can modify the surface is by producing fusion proteins. So you take proteins like tetraspanin, and then you retain the transmembrane structure, and then you include For the extracellular structure, the protein of interest, it could be an antibody, it could be a receptor of some kind that targets your EV to your target tissue. Adhesion molecules are a similar story. One interesting molecule that a lot of people are working with right now is CD47. For those of you who don't know, CD47 is a very ubiquitous cell surface molecule that instructs macrophages not to phagocytose the cell. So that's the don't eat me signal. And the reason for that is if you express CD47 on the surface of EVs, this hopefully increases circulation time by preventing them from being uptaken by macrophages. The caveat to that is we don't necessarily know the role of macrophages in the therapeutic effect, and perhaps uptake by macrophages is part of the therapeutic effect that's seen with EVs, especially in regenerative medicine, but that's a different story for a different day. And then finally, the lipid content can be modified by supplementation of media and other approaches. So as far as EV drug-based, EV-based drug delivery is concerned, the rationale behind it is that they're natural paracrine shuttles. They're biocamouflaged, so they're less likely to be fixated by albumin or complement, although that does happen. They're able to traverse biological membranes, so-called immune-privileged biological membranes like the blood-brain barrier and other parts of the body like the kidney. Using EVs circumvents lipid nanoparticle toxicity, which is fairly pronounced when you look at the RNA therapeutic trials, some of which have ended because of toxicity issues. And then also the possibility of targeting tissue by designing or at least choosing where that EV comes from. And of course, the cargo we've discussed before, they can be RNA therapeutics, they can be small molecules like doxorubicin and other chemotherapeutic agents, they can be vaccine antigens, and they can be proteins like growth factors and the like. And I wanted to show you a very interesting talk that was delivered a couple of weeks ago in Bethesda, where a group out of Italy, Professor Giovanni Camusi and others, were able to design an orally available EV vaccine from orange juice. So they isolate EVs from orange juice and they transfect mRNA from the spike-1 protein of coronavirus, and they give animals either orally or intranasally. And as you can see, by the end of 5 weeks, these animals have increased IgG circulation in the blood, increases of IgA in the alveolar lavage, and an increase in spike-1 neutralizing antibodies in the blood, showing that delivery by oral or intranasal approaches of this transfected EV is therapeutically efficacious. This is another example of EVs derived from dendritic cells and loaded with doxorubicin, injected in an ectopic model of breast cancer tumor. And as you can see, the EVs are capably transmitting the drug to the tumor site. In this second part of my presentation, I'll focus on EVs in regenerative medicine. And from a historical standpoint, the way that we found ourselves to EVs was the realization that many of the cells that were being used, whether they're MSCs or CPCs or CDCs or any of the cell therapies that have been tested, they're very transient. They only last a couple of weeks after injection, and then they're gone. And yet the effects that they have are enduring. And we realized that it's really more the secretome, and more specifically EVs, that are uptaken by certain cells in the injured microenvironment that induces these effects. And what you see here is one of the first papers to describe the role of EVs, and in this case from Professor Lim's group, showing EVs from MSCs being uptaken by smooth muscle cells. And then on the right, this is very early work that we did 4 years after, showing that EVs were both necessary and sufficient to mediate the effects of cardiosphere-derived cell therapy. And this is looking at the echocardiography of mice given EVs. If you inhibit the ability of the cells to release EVs, then as you can see, this red line, cardiac function decreases over time, whereas with CDCs that are capable of secreting EVs, that function is retained. And if you inject CDC EVs in place of the cells, then you're able to recapitulate the full effect, whereas EVs from fibroblasts are therapeutically inert. The logical step then is to mine the cargo of these EVs and ask, well, what effects are these signals having? And for the better part of 2 decades, we and others have focused on microRNAs. And what you're seeing here are all the different cell types that have been evaluated, or the EVs from all the different cell types that have been effective in cardiac injury. And all we're seeing is microRNAs. And that's all fine and good. It's easy to study microRNAs if only because we understand the mechanism. But that's almost like looking for your keys in a dark alley only under a lit light post, right? And so the one other sort of discovery that many groups have been making is that microRNAs are few and far between. In fact, some microRNAs are either 1 copy per 100 or 1,000, sometimes even 10,000 particles, which makes us wonder if they're actually playing a therapeutic role at all. And that's pushed us to look at other RNA species, which are probably more abundant, but that takes us down a road where we need to understand the mechanism of these previously undescribed RNAs. So the current challenges with EV therapy are the same ones that we have with cell therapy, and that is a fundamental lack of understanding of the mechanism of action. What are the pathways that need to be preserved in therapeutic cells for them to secrete therapeutic EVs? And for EVs, what cargo do they have that are necessary and perhaps sufficient to drive the effects that we're seeing, and what are the target tissue? Those enduring questions are the reasons why someone was just talking a little while ago about the lack of reproducibility in research. Well, in this case, it's lack of reproducibility in clinical trials, especially going from phase 1/2 to a phase 3. They have very promising results early on, but those results become lukewarm as you approach phase 2 and 3. Some people say, some experts say it's because of the scale up. Others say that it's because we just don't know what to control and what to conserve. And the fact that primary cells are by their very nature inconsistent. So that accentuates the importance of finding out what this mechanism is. And I'll just give you an example from our group. This is an ELISA of many different CDC donors, showing that the content of beta-catenin, the levels of beta-catenin in CDCs correlates very strongly with their therapeutic efficacy, at least in a mouse model of myocardial infarction. And so we chased after that pathway, and we were able to engineer CDCs and EVs since then. But of course, this is work ongoing. So I see Mario approaching. So the conclusion of this talk is that EVs possess properties that uniquely position them as therapeutic agents, and those applications include drug delivery, as well as therapeutic cell-derived agents. EV cargo and tissue targeting can be engineered, as we discussed, either through chemical manipulation of parent cells or EVs, or through genetic modification of the parent cells themselves. And that one of the enduring challenges, both with drug delivery and with regenerative medicine, is understanding exactly what it is about those cells or those EVs that produce the salutary effects that we see. This is the end of my presentation. I'm happy to provide free copies. This is my email. If anyone has any questions or wants to get involved in applying the EVs to therapy, we've learned through the School of Hard Knocks, so we're happy to share our experience with you, especially the graduate students who are just starting their theses. We're more than happy to help, and you're all more than welcome. Thank you.